Great. Well, thank you very much for the introduction, or not the written introduction, the invitation. I'm not from the West Coast, but somehow I didn't sleep very well last night, so you guys will have to. Maybe it's just because I grew up on the West Coast, so I have some sort of affinity to you guys. And right. So so today I'll tell you about some of the ideas that we've been excited about in my group. So we're in the physics department at MIT. Our general approach is to use these laboratory microbial populations to explore some fundamental ideas, either in theoretical ecology or evolutionary dynamics, and you'll kind of get a sense of what I mean by that over the next half hour. Right, so I'd say that the talk will really focus mostly on this question of how you can anticipate sudden transitions or tipping points in biological populations. The idea is that natural populations can collapse suddenly in response to deteriorating conditions, and we'd like to anticipate that in advance. The general reason that such populations can undergo these transitions is because of positive feedback loops between members of the population. And in this case, the cells that we're studying are kind of collectively breaking down a sugar. And in that case, you always have to ask, well, what happens if one of those cells stops producing or paying the cost to make that public good? And we've done various experiments looking at the evolution of cooperation with this as a model system. All right, so perhaps the most famous example of a population collapse is that of the Newfoundland cod fishery off the eastern seaboard of Canada. So what's plotted here on the y-axis is the number of fish that are caught as a function of time. So what you would really like to have here is population size, but we plot the data that we have. Right? And what you can see is this is down 1850 up to modern era. And this fishery really provided the source of livelihood for kind of tens of thousands of fishermen and their families for hundreds of years. But in the 60s and 70s, improved fishing technology led to this dramatic increase in the number of fish that were caught, which in the early 90s led to a kind of a dramatic collapse of that fishery. Okay, so even though we have not been fishing here for the last 20 years, only now are there kind of tentative signs of recovery. Now, this basic phenomenon, these collapses can occur because, as I said, you have, in many cases, individuals in a population benefit from the presence of other individuals. Right, so for fish, this, of course, happens with schooling behavior. If you don't have enough fish, then you can't form an effective school, and you all get eaten. But this is more general than just fish populations. Of course, animal populations also engage in herding behavior. Wolves engage in group hunting. And many of us in the room are familiar with many examples of cooperative behavior in microbial populations as well. And we're going to be talking about one example of that in a moment. But before we get there, I just want to kind of explain why it is that these sudden transitions can take place and how to maybe think about it conceptually. So what's plotted here is going to be a bifurcation diagram where on the y-axis now we really are going to be looking at population size. And on the x-axis is some sort of environmental condition with deterioration on your right. So what you can see is that as the environment's deteriorating, there's some sort of modest decrease in the population size, but nothing that you'd say is especially alarming. However, at some critical environmental condition, this could be some critical number of fish that are caught in this fishery, what can happen is there can be a collapse of that population. Could be local extinction or transition to some other undesirable state. Okay? So not only did this happen suddenly, but it may be difficult to reverse. Because even if you issue fewer fishing permits, it may be the case you have to come all the way over here to the left before you get recovery of that population. Okay? So what happened here is that you have two stable states that are separated by some unstable state here, denoted in the dash by a dashed line. Okay? So that unstable state tells you, depending on your initial population size, which of those two stable states you'll go to. Okay? And this basic structure, it's called a fold bifurcation. And it's what is thought to underlie sudden transitions in a wide variety of complex systems. All right? So in populations, ecosystems, but also researchers study this in kind of climate regime shifts, financial markets, all sorts of different things. And in this field, then, the question is, well, how is it that we could have anticipated that tipping point was going to approach? Is there any early warning indicator of that impending collapse? Because what you'd really like to know is that you're kind of approaching the tipping point before you cross it. Because after it happens, it may be too late. Okay? What's interesting is that from nonlinear dynamics, there are, in principle, universal early warning indicators associated with these transitions. To get an idea of why that might be, what we're going to do is we're going to look at this bifurcation diagram, and we're going to kind of take two slices from it. The green slice is an environmental condition that's far from the tipping point, and the red slice is when we're close to it. And now what we're going to do is we're going to kind of rotate this diagram. So now the population size is going to be on the x-axis, and we're going to plot an effective potential on the y-axis. Okay? So what we have here is in this place where we're far from the tipping point, you can see that the population is in some very nice potential well. Okay? And in particular, any sort of perturbation quickly will go away. Right? However, as we approach the tipping point here, F2, what you see is that the effective potential looks very different. Because now there's a very kind of broad potential. It tells you that a perturbation in the population is going to take a long time to go away. And that's, this effect is called critical slowing down, because recovery from perturbations becomes increasingly slow. All right? Now, this is sort of an intuitive notion of what's going on, but you can, of course, formalize this mathematically. And the statement is that at these bifurcation points, the dominant eigenvalue goes to 0. Right? And this should manifest itself in an increase in this return time. But even if you don't have a concrete perturbation, it should lead to a change in the fluctuations of the population, because fluctuations here will get slower and will get larger. Okay? So the idea here is that there should be kind of two universal, hopefully, early warning indicators associated with these transitions. And you should be able to measure them before you reach the tipping point. Right? So in particular, the fluctuations should get larger, and they should also get slower. All right, so this is, I think, an interesting idea. And the question is whether it can be measured in kind of real living populations. So this is the task that 2 graduate students in my group set for themselves. Allee is a physics graduate student. Don Vorsland was a visiting master's student from the Netherlands. And what they decided to do is use these yeast populations in order to measure these effects in the laboratory. Right. What you need to know about our yeast for now is just that when yeast grow on the sugar sucrose, they do so in some sort of collective fashion, because the sucrose, that sugar, is broken down outside of the cell. Right? So just like many people are going to be talking about, the yeast secrete this enzyme that breaks down the sugar, and that allows the entire population to grow. And this manifests itself in a very concrete way that the yeast actually divide more rapidly at higher cell density. Of course, eventually, the yeast start running out of resources, and the division rate goes down again. But this effect is called the Allee effect when, in the context of population dynamics. This leads perhaps to this possibility of sudden collapse, although we have to see if that really does happen. Okay. All right, so the experiments are rather simple, so I just want to be clear about what it is we're doing. All right, so what we do is we start at some low cell density in well-mixed batch culture liquid. We shake at 30 degrees, let them divide over the course of a day. Now, at the end of the day, what we can do is we just measure the cell density by looking at the optical density of that media. Right? Then what we do is we dilute by some amount, daily dilution, where we transfer only a small fraction of the cells to fresh media, fresh sucrose, for example. And we repeat this procedure for a week or two. Right? And then we just look to see how the population size at the end of each day varies over time. Okay. Now, the data I'm going to show you in a moment, for example, we're going to start with a daily dilution of 1,400. Okay? So the cells kind of have to divide for 10 or 11 times over the course of the day in order to keep up with this imposed death rate from the daily dilution. Okay. Now, the first question that we wanted to ask is whether it's really the case that yeast growing this sugar sucrose display bistable fates depending on the starting population size. Right, so this is something that is that's difficult to confirm with animal populations, but with microbial populations, it's kind of trivial. Right, so all we do is we start with different population densities, and we just propagate daily dilution over time. And what you see immediately is that the fate is indeed bistable. Right, so if we start with a large density of cells, those populations reach some stable equilibrium. But if we start with a low density of cells, those populations all go extinct. So that's another stable state. And those two stable states are then separated by some unstable state in between. And indeed, what you can see is that these 8 populations that were all started at a nominally identical cell density display kind of stochastic fates, because some of those populations look like they're going to survive, and some look like they're going to go extinct. Okay. All right, so we can take this data, for example, and then plot that location of the stable state, unstable, and then stable again down at 0. And this is density as a function of the environmental condition. Dilution factor was 1,400, so we have the stable, unstable, stable down at 0 again. Right? What we would really like to do, though, is to see if yeast growing on this sugar have a fold bifurcation, where those stable and unstable fixed points kind of come together and collide and go away. Right? Once again, we can do this with microbial populations because we just repeat that previous experiment, but with different dilution factors, different environmental conditions here on the x-axis. And when we do that experiment, what we get is, I think, a really beautiful fold bifurcation diagram describing how yeast grow on this sugar, where as the environment deteriorates, we get a decrease in the stable population size. The minimal population size required for survival goes up. And the tipping point occurs when those two fixed points collide. Okay. All right, so now we've confirmed that this population indeed has a fold bifurcation. I think that's important because in this field, many people have models suggesting that their lake or their bird population, fish population, have such a fold bifurcation, but nobody had actually measured it in a living population. Right? But now that we yeah.
When you say population density, do you mean the final population?
Yeah, so everything that's plotted is the population size at the end of the day. Yeah, so then you can then calculate, for example, the population size at the beginning of the day if you want as well from the. Yeah, so the beginning of each day, we start with the same amount of sucrose. Yeah. Right. And the idea here is just that you have to have enough cells to break down the sugar so that the cells can, yeah.
Since you break it down, then there will be more population size. Right? It seems to me it should be kind of more or less the same. I don't understand why you would even get to the middle, the lower cell density.
Why you would get here.
Yeah. So if you see the starting point is probably much lower than that, right? So for example, the starting point is probably much lower than that.
Well, we in these experiments, we can in principle start with any density of cells that we want, and then look to see what happens over time.
Right. So the top stable is very obvious, right? But the unstable, these points.
Yeah.
If you have waited longer, like use those blue lines right above the unstable fixed point, if you just wait longer, would they actually go to the stable?
Yes. Yeah.
So this will be counted as stable.
Right. So the idea, yeah. So it's really this is the stable state. These just have not reached there yet.
Right. So it seems to me there should be only one stable state, right? Whatever is limiting it by the.
Right. So there's really one stable state up here, but then there's also a stable state down at extinction. Whereas you can see that these guys really are going extinct. So 0 is also a stable state here.
So there are two. But I don't understand why you see all these intermediate values.
Oh, these are just right. So the idea here is that there's a from the standpoint of the potential, there's a potential energy maximum here. So it's in principle, if from a deterministic without noise, if you sit right on that unstable state, you won't move. Right? And equivalently here, what it means is that it's kind of like a random walk here. And it just takes a long time for those populations to resolve.
Population size at the end of the day, whatever that is, right? So
Yeah.
It's not like you wait forever until it really reaches stable. Just a potential.
Right. So the idea here is that there's always going to be some death, right? So from the context of plant populations, for example, they send out their seeds, and then they grow up, and then there's a yearly cycle, right, where there's some death rate. So it's the same kind of idea that there's some death that's imposed on the population. Yeah.
I guess here the idea is that at the end of the day, the measurement here is that it's not too far away from your anticipated fixed point. Is that the assumption? That's the assumption there, right? If it's not true fixed point, the fixed point is unstable. Right? Just wait long enough.
Well, I think you can have unstable fixed points. I would say.
Sure, but it's unstable. But if experimentally, if you wait long enough, it can bifurcate. So either they are.
Well, and that's indeed what's happening here. Yeah, no, I think that you could, and all this could have been done in a chemostat as well, in the sense that here it's just that we're doing dilutions once a day instead of continuous dilutions, right? So it's just there's some world that these cells are living in, and then the question is how does the population size change over time? Does that? Good one. This is population density at the end of the day. Yeah, so this is at a fixed and
interrelate somehow, right? So dilution factor
sure.
gives you the population density starting each day.
Yeah. Yeah. So if you wanted to, you could have plotted the population density at the beginning of each day,
Right.
and you can see how to calculate that from.
But basically, does the initial population density matter at all? Or
Yeah. So the initial population density is also changing.
Or something, but then the effects die out, right?
I think it's just like any other dynamical system, in the sense that there's some potential, and you could do this from the standpoint of here as well, right? So you can start anywhere you want, and you can just look to see where does the population go, and eventually, here you come to some stable state. And so when we're doing those experiments, we're just starting at many different population sizes so that we can see, for example, here there's. By doing many population sizes, you can say, all right, well, here's a stable state. Maybe over here is another one. Here's an unstable fixed point as well. And just you can look to see how the populations kind of, how they evolve over time. Although it's dangerous to use that word because it means something different in dynamical systems than in population. Yeah.
Is it possible to illustrate this concept in this experiment that you have actually just illustrate this concept of mass distribution?
Yes. So the idea is that here we start with many different population sizes. All the populations above this population size come over here. The population sizes below come over here. Right? So that's what we mean by bistable.
But to show that with the
Yeah, we also have a model that of yeast growth in the sugar that explains all this. And indeed, actually, the line that I showed there was actually from our simple kind of growth model of yeast. So yeah, so actually, yeah, a simple model of yeast growth actually leads to this structure. Yeah, so I'd say that we think we understand how the yeast are growing in the sugar.
So the idea here, again, I'm still trying to understand the basics of this experiment. So at the beginning, you have a certain starting density near the unstable fixed point, right? And
That's
at
right.
the end of the day, the fluctuation is not sufficiently large because it's low fluctuation.
Right.
With that particular day, the population still hasn't either bifurcated to the upper branch
That's
or
right.
lower branch. Then you use that basically saying, okay, this is our sort of experimental surrogate of the unstable fixed point. Is that good way of?
Right. I mean, I think that you could. I mean, from this data, you can kind of just see where the unstable fixed point is, right? That you can see that these populations are just not moving very much right in here. But once they get away from this particular density, then they really start moving deterministically. Right? Does that make sense? All right, so we have a fold bifurcation, which is great. So now we can start doing things with it. Right? So one basic kind of prediction from theoretical ecology is that as populations move here to the left, as they are experiencing more challenging environments, that they should become less resilient to perturbations. All right, so to explore that idea, what we did is we just allowed populations to reach equilibrium, and then for one day we just applied a salt shock. Right? So it's just not quite as happy of an environment, just look to see how the populations respond. Right? So in a benign environment, you can see that the population size kind of dips, but then rapidly recovers. In a more challenging environment, now the population size goes further down, and now it's taking longer for the population to recover, even after going back into this original environment. Right? So you can already start to see some signatures of critical slowing down. Right? There's an increase in the recovery time as the environment deteriorates. If the environment gets worse, now you can see, oh, some of those populations may survive, some may not. And in even a more challenging environment, those populations are all going extinct. So what you can see is that we can experimentally observe a decrease in the resilience of the populations as we move to more challenging environments. Yeah.
Just do they evolve actually when you do this?
Yeah. So most of these experiments are done over time scales where mutants can arise but maybe not have time to spread. In your experiments, there's sort of severe selection to try to improve things. Yeah, but we've actually done experiments also over the course of a month, and we can actually still see a lot of these indicators. And so the behavior is robust, even if we do allow evolution in the population. Right. OK, so now the question is, well, is it really the case that populations, as they approach this tipping point, is it really the case that the fluctuations change? Can we measure these proposed early warning indicators? Well, first we can look at the size of the fluctuations. So this is the coefficient of variation, the standard deviation divided by the mean. What you can see is that in benign conditions, fluctuations are only a couple %. However, as we deteriorate the environment, then the fluctuations increase dramatically. Yeah. This is the fluctuations in the population density from day to day.
So you just use the OD.
Yeah. Right. So this, I think, in our case, is the proper kind of metric for noise in the system. But even if you just look at the raw size of the fluctuations, just the standard deviation, even that increases in the system, despite the fact that the population size is going down. Okay. Yeah.
I hate to interrupt you.
Yeah, that's fine.
But so here, the number of cells per volume goes down.
Yes.
Wouldn't you expect from just sheer number idea that you have lower something fluctuations a little higher?
Well, OK, but now you're thinking about because of root end fluctuations, right? So you're saying that the percentage error maybe should go up. But what I'm showing here is that even just the raw size of the fluctuations goes up. Right? So you would the kind of physicist's intuition is that fluctuations should be at root n. Although in our case, I think we have linear n scaling of noise. But even from that intuition, you would expect that the proportional noise might go up, but the absolute value of the noise should not go up. That should still go down as the population size goes down.
All the distributions of these
Yeah. All right. Well, I'll show you some raw data in a moment, just so you can. All right. So let me just say that we can. All right. So this says that we can use the size of the fluctuations as an early warning indicator that the tipping point is perhaps going to approach. What about this other indicator, the time scale of the fluctuations? Well, once again, for that, we can look at the autocorrelation time, once again, of the population size fluctuations. In a benign environment, typical fluctuations go away in less than 2 days, whereas once we approach the tipping point, then the fluctuations last for about a week. Okay, so once again, the fluctuations get they increase in size and time scale before the tipping point. And that's in accordance with the predictions from this theory. Right, so it's good to look at raw data always. So this is kind of plotting the deviation from the mean on one day compared to the next day. So this is population size on one day and the following day at a benign environment and a challenging environment. So the idea here is that you can just see that the fluctuations, first of all, they increase in size. Right? This is the same scale here. But also, the fluctuations become more correlated as the environment deteriorates. The day-to-day fluctuations in population size fall more along the diagonal as we go to these more challenging environments. And this is just another way of saying or visualizing this increase in the autocorrelation time. Right? That a large population on one day tends to be large the following day in challenging environments, but not so much in the benign environments. All right. So there are other questions. I have a list of questions here, but which are not actually any of the questions that you guys asked, but I'll go over them anyways. All right. So you might ask, well, can these indicators be observed in a continuously deteriorating environment? The experiments that I showed you, what we did is we just took these populations, 20 or 30 of them, and just placed them in different environmental conditions and looked at the fluctuations. But in practice, what you'd really like to do is continuously deteriorate the environment and see if you can measure the indicators. And the answer is that we can. Right? So as I kind of mentioned, over the course of 3 or 4 weeks, we can slowly deteriorate the environment, and we can actually see these indicators increase before we cross the tipping point. I think it's also interesting to ask about other ways of deteriorating the environment. What we'd really like is to be able to use these things for natural populations where we won't even know how the environment's deteriorating necessarily. Right? So to explore this idea, what we've done is instead of increasing this dilution factor, we just decrease the sugar concentration. And indeed, we can still see an increase in the indicators. Regarding the distributions, it's also been proposed that the skewness of the fluctuations, the asymmetry of the fluctuations, should increase as we move towards the tipping point. Interestingly, we do not see any evidence of this. And I think this is part of the value of laboratory experiments, where we have a controlled situation where we can clearly see some indicators, but we don't see others. Right? And so this could provide some guidance to researchers studying these phenomena in natural populations.
Did you predict the skewness of the fluctuations?
Did we predict it? You're saying? Yeah. So actually, our model actually, from our model, you would expect that the skewness would not be a good indicator either.
Okay.
Yeah. But the model doesn't necessarily have to be true either, right? But it does make us think that we know what's going on. Yeah.
I'm not sure I understand this last statement, because if you really have barrier to steady states, right?
Yeah.
Lowering itself down, right? Then I would expect that the deviation towards the
Yes. Right. So yeah.
one is going to be longer, that one is stay for longer, and so on and so forth, right?
And you're saying, but now you're saying that the skewness.
I would expect skewness, right?
Yes, right. So exactly. So the reason that you might expect the skewness to increase is because this effective potential, as it's bending over, it becomes asymmetric, right? I think that in our case, what's happening is that at these benign environments, you're really running into the into carrying capacity. So you have kind of the one wall really goes up. I don't know if I'm going to be able to draw anything that this. Right. So the idea is that it.
Yeah, there.
Okay. Right. So the idea here is that it's really steep on one side in this benign environment, whereas in a more challenging environment, this might curve over, but you also kind of the other side of the potential is also changing, right? And this just highlights that the change in the skewness is not universal in the same sense that the eigenvalue behavior is. All right, and you might also expect that in natural populations, which are spatially extended, that in addition to this critical slowing down in time, right, that you can look at changes in the behavior in the temporal domain, you might also be able to see changes in the behavior over space as a function of position. And indeed, we have a recent set of experiments exploring this, and we've been able to see spatial patterns before it collapsed. But I'm not. I want to talk about some other things. I don't know. I might be close to running out of time, anyways. But yeah, so I'm not going to talk. I'm not going to talk about this more, but I just want to mention it in case anybody's curious. I'd be happy to talk about it later. Yeah, so I just wanted to end with thinking about some of these experiments looking at the behavior of cheaters. In particular, the reason that these populations display a fold bifurcation that have these kind of sudden collapse type behaviors is that the yeast cells are collectively breaking down the sugar sucrose. So you have to then ask, well, what happens if one of those cells stops expressing that enzyme, right? So now what we have is we have this population of yeast. They're all happily growing on sucrose. What happens if one of them stops making that enzyme, right? Well, if all of that sugar, the public good, is shared among the population, that sugar should spread throughout the population and indeed should have an advantage at any fraction of cooperator or cheater, right? Now, in that case, it's always better to be a cheater and it leads to the extinction of cooperation. Okay? However, experimentally, as a postdoc, I worked on this system that was motivated by work by Greig and Travisano. And what I found was that actually we get coexistence between cooperation and cheating in the system. And that's basically because the cooperator yeast cells, they have some preferential access to the public goods that they create, because some of that enzyme gets stuck in the cell wall close to the cell. Right? So in this case, we actually have coexistence between cooperation and cheating. Right? So we've been using this now to look at some other kind of dynamics, really at the edge of evolution and ecology. I'll give you kind of some examples. I'm just going to highlight some other work that we've done so that if you want to ask questions later. All right, so one is that we've found that if you add a bacterial competitor to these yeast populations, that actually drives cooperation within the yeast in the sense that the cooperative fraction increases dramatically as a result of this bacterial competition. Okay. We also have done experiments looking at range expansions. So what happens if you have these cooperative populations expanding into new territory, and we find that this actually also favors the cooperator genotypes. Okay. So yeah, I'm going to be around the next couple days if you want to chat about any of these things. But I wanted to close by telling you about some recent experiments that my postdoc, Alvaro Sanchez, has done exploring coupling between population dynamics and evolutionary dynamics. And I think that his data are very pretty, so I wanted to share it with you. All right, so the motivation of this is that we often think about evolutionary dynamics and population dynamics as occurring over very different time scales, in which case we don't have to consider them interacting in any kind of crazy way. Right, so if you think about evolution, it's very slow, whereas if you want to understand, let's say, the population dynamics on Easter Island, this occurred over a shorter time scale, so you don't have to think about changes in the evolutionary kind of state of the population in order to understand why it was that Easter Island population collapsed. Right? Instead, you just assume that the evolutionary state of the population is fixed, and you look for other factors. Right? So maybe they, I don't know, Jared Diamond has theories on this, and you can read his book. Okay? Right, so, however, in microbial populations, selection is often very strong, and that can lead to an overlap of these timescales. Right? So let me explain some just very simple experiments that Alvaro did to try to get at this. All right, so at the beginning of the talk, I told you about the population dynamics of these yeast growing in sucrose when all of the population are cooperators. Now, what we're going to do is show data where we instead just do these daily dilutions, but allow for some mixed population of cooperator-cheater. And then I'll show you what things look like. In my original experiments, I had fixed population size, so there was no potential for coupling here. Okay? All right, so if you just look at the population density over time for various cultures, what you see is that some populations go up and then down, down, then up. All right, they kind of do all sorts of funny things. Okay? Whereas if you take those same populations and you look at the cooperator fraction, this is as assayed by flow cytometry, what you see is, again, things go up, down, up, left, right. All right, all sorts of funny things. Right? So you might just look at this data and say, well, biology, it's hard, it's messy, it's complicated, and then you could just go back to studying kind of solid-state physics. Right? But what's interesting is if you take this exact data and you plot it in a different way, then you start to see some really intriguing patterns. In particular, if you plot the evolution on the y-axis, population dynamics on the x-axis, what you see are some really striking things. Right? So this red population is indeed going extinct, but these other populations seem like they're spiraling in this space. Right? So this is kind of a direct signature of what is known as ecoevolutionary feedback. Right? So a case where you have interactions between an ecological variable population size and an evolutionary variable, this allele frequency. So what happens here is that you start here. There are not enough cooperators to break down the sugar. Population size starts going down. But that starts favoring the cooperators because they have some preferential access. Cooperative fraction starts going up. Right? But as that happens, now there's more and more sugar, so the population size starts increasing. Okay, but as that happens, now that leads to a situation that favors the cheaters. And so then you get a decrease in the cooperative fraction. So this leads to some spiral in this ecoevolutionary space. And indeed, the simple model that I told you about, I'd be happy to tell you about it more. It predicts that this basics that you expect the space to look like this. So that all the dynamics at the beginning of the talk were occurring on this f equal to 1 axis. Everybody's a cooperator. Stable state, unstable, stable again down at 0. But the real evolutionary dynamics are much richer, because you have this separatrix here that separates a region of the kind of populations that are going to go extinct, and a region here that, that spirals to coexistence. And indeed, if you just take a bunch of populations, track them over order 100 generations, what you see is that things really kind of look like this. All right? Small n's are kind of noisy because it's hard to measure those accurately. But what you can see is that there really are some beautiful spirals in this space coming to a state of coexistence. Right? And indeed, what's interesting is that the presence of these cheaters doesn't significantly reduce the population size. But what we found is that it does reduce the resilience of the population, both to perturbations and to rapidly changing environments. All right, so in the last, do I have a few minutes left? 2, all right, perfect. All right, so let me just tell you what happens here when these populations are pushed towards a point of collapse, just to kind of bring it full circle to the beginning of the talk. All right, so now what we have are kind of 2 players in this ecosystem. We have like a producer, the cooperator, and a consumer, this cheater. All right, so as we deteriorate the environment, the total population size goes down. But what's interesting is that if you look at the cheater population size and the cooperator population size, they do different things. So the cooperator density actually goes up. And so this is kind of weird, right? Because we normally think about population size as a 0th order estimate for the health of a population. Whereas here, you see the population size goes up before collapsing. Right? However, if you look at the size of the fluctuations, that increases for all 3 of these population sizes. So it doesn't matter whether you're looking at the total size, cooperator size, cheater size. The fluctuations increase for all 3 of these populations, telling you that this really does work as a good early warning indicator. Right? Now, you might also ask, well, what happens? So this is experimental data, kind of zooming in on those spirals coming into here, which I'm really very fond of, so I just wanted to zoom in on it. But the question is, how is it these spirals change as we move towards a point of collapse? Right, so now I'm going to switch to what our simulations argue is happening, and we have data saying this happens as well. The idea is that as the environment deteriorates, those spirals become more spirally. So benign environment comes here very rapidly, but as the environment deteriorates, the spirals get to be like this. And eventually, you get to kind of neutrally stable orbits, and then that's the point where the populations are going to collapse. Okay. All right, so that's simulations, just to be clear. All right, so what we did is we used cooperative growth of yeast in the sugar sucrose to study this phenomenon of catastrophic collapse. And we found that both the size and the time scale of fluctuations increase as you approach the tipping point. We also found that these cheater strategies that don't contribute to the public good can invade and take advantage of a population of cooperators, but we get coexistence and survival of the populations. And finally, we've recently been excited about this idea that we can get a lot of insight by looking at these ecoevolutionary trajectories of those populations. And I will end there just by thanking the group once again and funding sources. Thanks. All right.
Yeah, so with the fluctuations near the collapse point experiments, we did temporal
Yeah.
fluctuations. Have you tried an ensemble, meaning spatial? And the reason is because I'm feeling that with temporal fluctuations, you kind of miss the populations that would go extinct. But with spatial ones, some may go extinct.
That's right.
You see a skewness in that.
Yes. So you. There's a lot of work that goes into trying to understand the behavior of spatially extended populations and extinction, and whether it helps, hurts. And it actually depends upon the nature of the forcing and so forth. So in the experiments that we did, this is kind of a picture of how we think about this. Critical slowing down says that it takes longer to recover from a perturbation in time, and that could show up as an increase in the fluctuations. Right? But what we've shown is that there's an increase in the kind of length scale for recovery. So at a boundary between regions of different quality, then you have to go further away from the boundary in order to get recovery of the population. Right? So that's the basic.
Actually, instead of actual space, I was talking about repeat experiments.
Oh, yeah. So in that experiment, actually, yeah. So what we actually did was we had like say 24 replicates going for about a week. So we then can analyze it both within a population and across replicates. Yeah, and so. Yeah, so we actually, the data is all shown in a region where none of them are going extinct, actually. Yeah. Should we? All right. Well, I'm happy to chat over coffee. So yeah, come on up. All right, thanks.